Good afternoon, everyone, and thank you for coming. And as Virginie said, I'm going to be telling you about love, loss, and lymphocytes, not necessarily in that order. So when most of you hear about chlamydia, you would think about two things. One is koalas, and the other is the sexually transmitted infection, which is not really surprising because chlamydia is actually the most common bacterial STI worldwide. And what people tend to forget when they talk about chlamydia is that it's also responsible for trachoma, which is prevalent in Central and West Africa. So the one thing I want to highlight is, yes, chlamydia is an STI. Yes, it's treatable, but it can also increase HIV risk quite dramatically through inflammation. And just some interesting facts, it's very, very, very old. It was first reported in humans in 1807, but people couldn't really figure out what it was. They first thought it was a parasite, then they thought it was a virus. It was only identified as a bacteria in 1966, in the early days of electron microscopy. Many species can be infected with a chlamydia of some kind. So humans, mice, koalas, pigs, parrots, sheep, and so on and so forth. Not all are STIs. So not all of them are sexually transmitted. For human chlamydia, there have been more than 200 vaccine candidates, and they pretty much all failed. Most of them never even made it to in-human trials at all, except the current one, which is currently in testing, made by the SSI in Denmark. All right, well, with all those vaccine candidates, why don't we have a vaccine? Also, chlamydia is how old and why don't we have a vaccine? Well, it's not really a global health priority. The WHO is trying to push STIs now as a priority for vaccines, but chlamydia itself, not really. It causes infertility in women, mostly asymptomatic in men. If it caused erectile dysfunction, I suspect there would be more funding available. It's easily treatable. So, course of antibiotics, there's no antimicrobial resistance reported. So it's highly treatable, tends to affect mostly women, no funding. Most people use a small animal model. So that is mice, except it's a different species of chlamydia. And so they don't quite have the same pathology. And also, mice are not tiny people. So if you develop whatever treatment or intervention in mice, it's not quite translatable, which is why we have so many vaccine candidates, but no vaccine. Vaccine antigens are also kind of tricky to choose. There was this one vaccine in the early days that was live attenuated, and they tested it in non-human primates, and basically the vaccine group did worse after challenge because of the inflammatory memory response. So obviously you have to pick your antigens carefully because you don't want that to happen. So there's a lot of hypotheses regarding immunity, and I'll tell you about some of them. There is the arrested immunity hypothesis, the partially protective immunity hypothesis, but we don't really know. And most of these hypotheses are from Canada and one research group, and that is because of the type of data they had access to. All right, so let's talk a little bit about those hypotheses. So first of all, immunopathology is caused by a sustained inflammatory response in the genital tract. In the eye, that causes trachoma, which is blindness. And one of the things that you can look at, if you see this graph over here, it is the cumulative incidence. Can you see? Yes, you can. So it's a cumulative incidence of pelvic inflammatory disease, which is one of the things that chlamydia causes, and in the number of years that it was done. And A over here, that last graph, is women who don't have any repeat diagnosis. And then B is those with one repeat diagnosis. C is women with two repeat diagnoses. So basically, as you go up, it's multiple infections. And you can see that the incidence of pelvic inflammatory disease basically goes up the more infections these women have. So infertility and PID tends to be associated with repeat or multiple infections. How would you measure multiple infections? So one of the things that you can look at is antibodies. Antibodies are associated with exposure, so the number of exposures, and you can look either in serum or in vaginal swabs, and you'll see that these antibodies are higher if somebody has multiple or persistent infections. So when people look at antibodies and say, oh, well, these people, these women have infertility or pelvic inflammatory disease, and they also have high antibodies, it doesn't mean that the antibodies are necessarily causing the inflammation. And so this is just the antibodies that are associated with inflammation. This one is particularly IgG, and this came from a meta-analysis. This is infertility, tubal factor, infertility, and ectopic pregnancy. And you can basically see the number of participants who were positive by antibody is quite a few. And if you look at the odds ratios, that is basically 1.95 and 3.5. So antibodies are quite closely associated with these negative reproductive outcomes. All right, why do we think there's some sort of partial protective immunity? So this graph is in Denmark, but it looks the same no matter which country you look at, if this data is available. And what you see is the incidence of chlamydia by age. You see that it obviously starts off quite low in the teens. Then you reach this peak in adolescence, and then it kind of tapers off. So older women tend not to have chlamydia. And this doesn't mean they're treated. It just is what it is. So this isn't necessarily an outcome of treatment. Also, because most infections are asymptomatic, like 90% are asymptomatic, again, people are not necessarily accessing treatment because they don't think anything's wrong. So we think the association with age and the fewer infections in older people is because of partial protective immunity due to these sustained antigen exposures. And then we have the arrested immunity hypothesis. So what they did in Canada is back in like the 70s and 80s, they were like, let's eradicate STIs. So they pretty much did a test and treat. And what you see here, so this is cases and this is the years. If you look now in the 90s, the chlamydia is here in blue and pelvic inflammatory disease is in red. So excellent, the pelvic inflammatory disease went down, but the actual cases of chlamydia went up, which if you're testing and treating, you don't expect it to go up, you expect it to go down. So what is happening here? So the hypothesis that was put forward is that because you're testing and treating, you're catching people quite early in infection. So because you're treating them early in infection, the protective or the partially protective immunity doesn't get a chance to develop. And this might not have much of an effect individually, but when you do that to a whole population, that might be the effect. So of course, these are all hypotheses. Okay, so now that I've talked to you about chlamydia in general, let's go on to my actual study. So this work is actually from my postdoc, which I did with Joanne Passmore, and they had a very nice cohort study called the Women's Initiative in Sexual Health, WISH. And they basically looked at 147 adolescent girls and young women, and they followed them for 6 months. And one of the first things that they saw was chlamydia. The chlamydia prevalence was actually a bit ridiculous. So don't worry about this all participants because there was more than one site, but I just want to highlight this is Cape Town for 149 participants, and the prevalence of chlamydia is 41.6, right? And the reason the study was done is because we wanted to look at STIs and other factors that could be responsible for HIV risk. Of course, nobody expected to see 41%. So one of the reasons we managed to get this 41% is because there was no syndromic management. So they didn't go by symptoms. Everybody who came in and enrolled in the study got the swab tested for STIs. And so if you do that in a population, turns out you can get up to 41.6. When we submitted this to actually anybody, they said, no, something went wrong with your testing. Nothing went wrong with our testing. We've actually sequenced that chlamydia and it's there. So this 41.6 is accurate. Okay, so we saw that screamingly high percentage of young women, adolescents with chlamydia. So we thought, okay, well, this is a really good opportunity to look at immunity to chlamydia because as I showed you earlier, we don't really know. We have no idea what's going on. We don't know how exposure history fits into immunity. We know how it fits into immunopathology, but what about immunity? What's happening on the other side? Uh, Also, we were very lucky because this cohort, because they're so young, they only have about 2 years since sexual debut, which means we are likely catching them at their first chlamydia infection, which is very rare in other cohorts. And so to do this, we looked at antibodies, right, as a measure of exposure. We looked at Th1 responses in PBMC. We also looked at genital tract infection and mucosal T cell activation. So that was also in the genital tract. So of these participants, I just want to highlight that they were median age was 18 and the age of sexual debut was 16. So it's only been about 2 years, right? So if they have chlamydia, chances are very high that this is their first infection. The other thing I want to point out is regular condom use in the last 12 months was quite terrible, 34%. And as I said, that chlamydia, even though the number of participants is a little bit different due to sample availability, it's still in the 40s. And another thing that will become important when we talk about exposure is whether or not they had a previous STI diagnosed or treated. And as you can see, 14%. So that's most of them didn't have a previous STI diagnosed or treated, which doesn't mean they didn't have a previous STI. It just means they weren't diagnosed or treated. Okay, so one of the first things we saw in this cohort is that multiple infections are common. So remember, this was a 6-month study. And so the participants were sampled every 2 months. And at baseline, 43% positive for chlamydia, 57 were negative. But then if you look at this positive, actually, if you look at all of them and you look to see how many positive chlamydia visits there are, most participants who had one chlamydia visit were still positive at the next visit. So the iffy thing about this is treatment. So they were all offered treatment. They were all given treatment. Whether they took the treatment or not, we can't say. They were all also given partner referral letters for their partners to get treatment. But again, this is not something that you can actually follow up on because if you ask them, of course, they're going to say yes. So take this with a pinch of salt. Okay, so now looking at exposure history, because we've got this lovely cohort of people who are close to sexual debut and are likely experiencing their first infection. How do we tease that out? People who have multiple infections, is that the same infection? Is that an old infection? Like, is there any way of doing that? So one of the ways of doing that is, like I said, serology. So that's antibodies. So we ran IgG on all of these 145 participants. 44 were seropositive, 81, so that's 56%, were seronegative, and about 20 in the process of seroconverting, or the other way around, right? We can't actually tell which way around that is. Now, if you put that together with the PCR diagnosis, so NAT is nucleic acid amplification test, which is just a PCR, right? It's just the diagnostic PCR. So if you put the serostatus and the PCR data together, you'll get something like this. So this is the entire cohort, right? And then you can break it down into those who were negative at baseline. So they did not have an active infection. Their PCR was negative. Okay. And most of those who were negative by PCR were also seronegative. Okay. So they never experienced chlamydia before. They don't have the antibodies either. 18% were positive and 10% were transitioning one way or the other. If you look at the PCR positive, nearly 50% of them were also seropositive. Some were seronegative, and a smaller percentage again were borderline. So one of the things that this is confirming in our cohort is that the antibodies are associated with active infection or antigen load, right? So you can see that the ones that are PCR positive, most of them have detectable antibodies. And if you look at the amount of antibodies here, the PCR positive, the NAT positive have higher levels of IgG compared to the NAT negative. So this is a nice little confirmation that, okay, when I'm looking at antibodies, it is a reflection of exposure. When you start thinking of combinations of the NAT and the IgG, what you can get is you can get participants who are negative for both. So those are uninfected and unexposed, right? They've never seen chlamydia. You get those that are PCR positive, so NAT positive, but antibody negative. So they haven't seroconverted yet. Seroconversion happens usually it takes about 6 months. So they've got an active infection, haven't seroconverted yet. You also have those who are positive for antibodies but negative by PCR. So obviously they had some kind of previous infection. And this previous infection, we think, is most likely cleared by itself because of that previous STI diagnosis and treatment was very, very low. And the untreated, recurrent, or persistent are positive both by PCR and antibody. So this sort of gives us a more nuanced view of exposure history. And just like I did with the PCR positive and negative, I also wanted to see if these three groups, these four groups, also hold up in terms of antibody and exposure. And you can see here, these are the PCR negative. And even though this one is antibody negative, right, it's obviously for antibodies, it's the lowest one. And then the next group is PCR positive. And even though it's antibody negative, the relative amount is still higher. It's still below the threshold, but it's still higher. So you can sort of see this is going up. So the antibodies are increasing with relative exposure. Okay, this takes us to our first interlude. So one of the first things you do when you want to measure immune responses is you say, okay, I have the samples, excellent, now I need the reagents. When I started the study, there were no reagents for chlamydia. So if I wanted to stimulate any cells with chlamydia, I had to grow it myself. Okay. And we did a fairly good job, I think. Look at this pretty picture. So this is chlamydia in green that's infecting a McCoy cell, which is a mammalian cell line, in red, right? And this is another picture just showing that, you know, we actually managed to get our protocol up and working, and it was great. And it basically involved lots of infections and blind passages, and this took a really long time. And I'm not really going to go through the steps, but they were, after we did the culture, we went through some lysis steps and some purification steps, including density gradient to separate out the chlamydia. And then to see whether we were successful, the only way to do that is to look under, is to use electron microscopy, remember, because this is so tiny. So over here is a heat-killed chlamydia. You can see it's still intact. And when you lyse it, it's gone, which means the lysis was successful. All right. And then my lesson in culture very quickly turned into a lesson in economics. The test that the microscopy test that we used to generate all those fantastic images was discontinued because there wasn't enough demand for it, because nobody uses it for diagnosis. Obviously, everybody uses PCR, and yeah, it was discontinued. So my poor master student at the time, her job was to find an alternative. So we tried other kits that claimed to be the same thing, and they also used fluorescent microscopy. None of them worked. We tried flow cytometry, both trying to look at the bacteria by itself and also intracellular. Didn't work either. We tried qPCR, but then we ran into problems because it's not a direct, you can't just take the CT value and say, okay, this is so many chlamydia CFU. That wasn't there was no direct correlation, which meant there was no kit available. So it needed a lot more fiddling than we had time for. So that died a sad death. So what did we do? We ordered MOMP E synthesized by GenScript, is what we did. Okay, so the interlude is over. We are now going on to talk about the T cell responses. So what we did is we have PBMC from all those women that I showed you earlier. and we stimulated it with recombinant MOMP. MOMP is the major outer membrane protein of chlamydia. It is the most immunogenic protein of chlamydia. And we looked at interferon gamma, IL-2, and TNF. And the way we chose this subset of participants is we picked people who had a positive PCR or were seropositive. Okay, so remember, there is a subset of participants who are seropositive but PCR negative. So if they were positive for chlamydia now or previously, that's what we looked at. So the first thing was, okay, let's do it the way everybody else does it before we start going into this deep dive on different exposure levels. So we looked at PCR pos and neg, right? And if you look at the CD4 response, so these are CD4 cells producing any cytokine in response to chlamydia, you will see that, okay, the negs look a little bit higher, but actually there's no statistical difference, which may be due to the small sample size. The sample size is a bit of an issue, but it still gives us some information. So we saw no differences in responders or the magnitude between the PCR positive and negative. We next broke them down into those three groups, right? PCR pos, antibody negative, PCR negative, antibody positive, and positive for both. So the exposure is going from those with a first infection to those who have recovered and those who have a current infection and probably a past infection. So we don't know if it's persistent, recurrent, repeat, we've no idea. But in terms of exposure, those are the most highly exposed. And when you look at that, you see that, first of all, if you look at the responders, which are those pies at the top, you see that responses are detected in all highly exposed women compared to 60% of those who are experiencing a first infection. And if you look at the magnitude, you see that the lowest response is those who are highly exposed. And I'm showing you the total cytokines. There were no differences in the individual cytokines. So the take home from this is that you can see that the CD4 T cell responses do actually reflect relative exposure here, right? Because you can see differences in the exposure groups. So in addition to looking at that, we also wanted to look at function. So these chlamydia-specific T cells, how many cytokines are they producing? What are they doing? So this is looking at cells that are making one cytokine, cells that are making two cytokines, and cells that are making three cytokines. And these are the different exposure groups. So that's just the same I showed you, exposure going that way. And the first thing that should stick out here is we didn't detect any polyfunctional cells in the most highly exposed group. They were not there in blood. And another very interesting thing is if you look at the one cytokine and the two cytokine producing cells, you basically see they look almost mirror images of each other, right? So the single cytokine producing cells are highest in women who are experiencing their first infection and lowest in women who are highly exposed. And you see the opposite for the two cytokines. If you were to look at it as proportions of each other, so in pie chart form, you will see that all of these look quite different. So again, exposure starts here. This is primary infection, this is recovered, and this is highly exposed. And again, you can see there's no 3 cytokine producing cells in the highly exposed. And you can see the proportion of single cytokine producing cells is also quite different between the primary infection and the previously infected. And just to point out here, which cytokine are they missing? Most of what these highly exposed participants produce is actually TNF, right? Which is pro-inflammatory and sort of goes to the more exposure somebody has, the more inflammatory it gets. So we also saw differences in polyfunctional responses based on relative exposure. So here you can see that everything is actually, we can link all the immunity here to relative exposure. So far, we used NAT and serology to identify 3 different levels of exposure. CD4 T cells in response to chlamydia were detectable in all highly exposed women versus about 70% in the other groups. But these responses in the highly exposed women were lower in magnitude, and basically they had no detectable polyfunctional responses. And the most polyfunctional CD4 T cells were actually found in the recovered group. So these T cell responses clearly reflect relative exposure. But okay, they do. What does that mean? So to try and figure out what that could mean, we looked in the genital tract. So previously, and this is quite well accepted, if you look at women who have an active chlamydia infection and those who don't, you will see higher concentrations of pro-inflammatory cytokines, multiple cytokines in the women with chlamydia. So chlamydia is generally considered inflammatory. That's not controversial. That's been quite well described. One of the things that is considered vital for pathogen clearance is interferon gamma. But, if you look at women with those persistent or repeat infections and many exposures, you will find that they have a higher concentration of interferon gamma in the genital tract, but they're still infected. So chlamydia has lots of immune evasion strategies that I'm not really going into. And a lot of its energy is put into evading this interferon gamma. The studies that said interferon gamma is vital for pathogen clearance were all mouse studies. So take that as you will. So what did we find? When we looked in the genital tract, we found an association between interferon gamma levels and antibodies. So that's showing exposure, which agrees pretty well with the literature. The other thing we wanted to look at in the genital tract is activated T cells. So is there any association between T cells that are infiltrating in the genital tract and exposure? Because remember, exposure is linked to immunopathology, which is linked to inflammation and activated T cells. So here we've got activated CD4 T cells, and again, you see a positive correlation with antibodies, which is also a measure of exposure. If you then, so this is for the whole cohort. If you then break it down again into those exposure groups, you will find that activated T cells are the highest in the most exposed compared to those who have recovered. And that probably is associated with antigen load, because remember, those who have recovered don't have an active infection, whereas the other groups do. So the exposure is very clearly associated with interferon gamma, with inflammation, and with activation. The next thing we did was to see whether the systemic immune response that we measured in PBMC had any associations with female genital tract cytokines. And what we found here, so at the top here are chlamydia-specific CD4 T cells, the responses in blood, and over here are all the cytokines that we looked at in the genital tract. And you see negative associations between CD4, interferon gamma responses, and IL-1 beta, TNF, MIP-1 beta, and IL-17. So not to overanalyze this, but the CD4 T cell response does appear to be associated with lower pro-inflammatory cytokines in the genital tract. And that might seem obvious, but again, most people do this kind of work in mice. So this is one of the first times this has been shown in humans. Okay, in summary, we identified, so the summary and discussion are sort of going to be together. So we identified 3 levels of exposure: primary, so those who are experiencing their first infection; recovered, so those who've had a previous infection but are not currently infected; and untreated or persistent or recurrent. We can't tell which one, but it's given the low treatment and previous testing, it's likely to be untreated. However, we did not take into account failure to seroconvert or anorectal infections. So the failure to seroconvert might seem like, oh, well, that's a big thing. There isn't actually any reported failure to seroconvert for chlamydia. These antibodies are detectable decades after infection. So the failure to seroconvert is possible, but again, very, very, very unlikely. And anorectal infections are again also possible, but usually they co-occur with vaginal infections. So if you picked up a vaginal infection, chances are you're also positive for anorectal rather than the other way around, at least mostly in women. CD4 responses were detectable in all untreated women. And that, of course, is probably related to the high antigen load. They're untreated, their PCR is positive, the antibodies are positive, meaning this antigen's been hanging around for a while. Okay, but we saw the responses were lower in magnitude with no polyfunctional responses. And we're not really sure why. It could be exhaustion just because of this antigen persistence. It could also be the cells, all these polyfunctional cells are maybe trafficking to the site of disease, the genital tract. We don't know. Nobody knows. It's just one of the things that we think might be happening. The other thing we saw was that CD4 responses were negatively associated with inflammatory cytokines in the genital tract and particularly interfere. So we think that interferon gamma in the genital tract, possibly from CD4 T cells, but others, may play a role in limiting immunopathology. All of these things are very difficult to tease out just because of the nature of the infection and the fact that, the infection takes place at a mucosal site rather than systemic. Okay, where do we go from here? So what have we learned during the study? We learned that both the magnitude and function of CD4 T cells are probably very important. And again, this might seem obvious, but people don't really study chlamydia immunity in people. Circulating Th1 cells may also limit genital tract immunopathology. So those are the two main findings of this study. But there's also food for thought. How do we disentangle immunity and immunopathology? How are we evaluating candidate vaccines? Right? Is it enough to evaluate these responses in blood? How do we develop a proxy for immunity in the female genital tract? So all of these are unanswered questions, and hopefully research will go that way. So thank you all for listening, and I'd just like to thank the Passmore Lab, the previous Passmore Lab, and all the funders and collaborators and study participants.